Hi everyone, welcome to my channel or welcome back. So in today's video I want to give part 2 of my late 2021 update on gene therapy. In the previous video I went over what's happening in gene therapies for different subtypes of limb girdle muscular dystrophy. And in this video I wanted to take a broader view on what's happening in gene therapies for other types of neuromuscular diseases, as well as some of the safety issues that people are looking into, and what some of the research goals are moving forward in gene therapy. So to start off the discussion on the current state of gene therapy, it's worth going over a few background points. So gene therapy as it currently exists puts a piece of DNA that contains a working copy of a certain gene that a person lacks or needs a good copy of into a type of virus called an AAV, which stands for adeno-associated virus. Now, this type of virus doesn't cause any human disease, and furthermore, when they're preparing this to deliver the DNA for the therapeutic gene, they take all of the virus's DNA out. So the virus can't replicate, can't infect anyone. It's just there to carry the gene that a person's body needs. But there's a few limitations. One is that AAVs are very small, which means they can only carry a certain length of DNA. That's not big enough to carry the full length of the gene for some diseases that we're trying to treat, and that includes the dystrophin gene, which is what has a mutation in Duchenne muscular dystrophy, DMD. It also includes the dysferlin gene, which is the gene that's mutated in limb girdle muscular dystrophy type 2B, aka R2, which is actually the type of muscular dystrophy that I have. Also, AAVs, well, ideally, if you're trying to treat a muscle disease, you want the AAVs to put all the DNA in muscle cells, or in some cases for neuromuscular disease in nerve cells. But what happens is the AAVs that are currently used often tend to wind up in the liver, which isn't where you want it. That increases the amount of AAV you need to give a person to get enough into their muscle cells, because not all of it is going there, which raises both the cost of producing that much AAV as well as concerns with toxicity, because you're putting so much of it in a person's body. In addition, the person receiving the AAV gene therapy, their immune system just sees, oh, there's all this virus coming into my body. This is an invader. I need to fight it off. And so the person develops antibodies to the AAV, which means you can't give them a second dose because their antibodies will attack the AAV before it can deliver the gene. So as it stands right now, AAV gene therapy is really a one-time treatment, although people are, as you would imagine, working on approaches to getting around that problem. So, in summary, and sort of this was best, put by Scott Gottlieb, who was the former head of the FDA, and who I, as well as one of my colleagues, got a chance to interview at the International LGMD conference a few months ago. As he put it, it's very early days for gene therapy. In the future, things will be much better. They'll be, much easier to produce, more cost effective, more therapeutically effective. So the technology is new and it's still being developed. That's something that, we need to keep in mind. A lot of these problems take a while to, be identified and then be worked out. The FDA had a workshop about 3 months ago in the beginning of September of this year that was actually streamed on the web, and I imagine that currently the videos of this are available on the FDA's website. So, and this was about the toxicity risks of AAV gene therapy specifically. So this went into some of the details. And so basically this was, looking into the data that's been gathered to date by the approved gene therapy for spinal muscular atrophy, as well as the current clinical trials for Duchenne muscular dystrophy, limb girdle muscular dystrophies, and X-linked myotubular myopathy, as well as some non-neuromuscular diseases. Okay, so hepatotoxicity just means toxicity to the liver. And this is a consequence of, as I explained a moment ago, a lot of the AAV that's given in gene therapy doesn't end up in the target organ. It actually ends up in the liver, which isn't where you want it. Now, the big problem that's occurred so far is in X-linked myotubular myopathy. There's been 3 deaths, as of, the time where this view graphs were. So one of the big safety concerns in gene therapy is hepatotoxicity, which means toxicity to the liver. And that's a consequence of a lot of the AAV that you give a person in gene therapy ends up not in the target organ that you want to deliver the gene to, but in the liver. And in particular, in the disease X-linked myotubular myopathy, even though it's primarily a muscle disease, it also tends to involve abnormalities to the liver. So there have been some deaths in this clinical trial, because the people already had pre-existing liver problems as a result of the disease, and then all of the AAV further compounded this issue. So this is very bad news for gene therapy for this particular disease. However, it's worth taking the bigger picture more generally, and while there have been some issues and instances of liver toxicity in spinal muscular atrophy, Duchenne muscular dystrophy, and limb girdle muscular dystrophy clinical trials, or in the case of SMA, in using the therapy after it had been approved, there have been no deaths. And in fact, the SMA gene therapy has been given to about 500 or 600 people by now, and with the prognosis of the more severe forms of those disease, a lot of them would not be alive today had it not been for the gene therapy treatment. So gene therapy is already saving lives, and that's a point worth remembering. So one of the complications in assessing liver toxicity in AAV gene therapy, which actually was inadvertently illustrated in some of the presentations and statements that the liver specialists in the FDA panel that I referred to earlier gave, is that some of the biomarkers which are typically used to assess a person's liver function are actually elevated in muscular dystrophies. So, which means that if you just measure that particular number, that this is either AST or ALT, or the abbreviations of the two biomarkers. A person with muscular dystrophy might appear to always have elevated liver levels of these, and therefore a liver problem, even if their liver is absolutely fine. So, what I'm hoping is that the FDA and also the drug companies developing gene therapies will realize this and, do the appropriate measures of liver function that won't be given false positives just because a person has muscular dystrophy. Okay, so the two major companies working on gene therapy for Duchenne muscular dystrophy are Pfizer and Sarepta, and so I want to go over a few recent details of the clinical trials from those two companies. So in the Pfizer trial, they found some what are termed serious adverse events, which are kind of the big red flags that the FDA looks for and other regulatory agencies look for in clinical trials. And what the company concluded about that is that in some cases, the people's body was having a immune reaction not to the AAV vector that's used to carry the dystrophin gene, but to the dystrophin protein itself, which is kind of ironic. It's exactly what the people's body needs, but since they've never been able to produce it before, their immune system recognizes it as being foreign. Now, for reasons that the company hasn't explained in detail, and so I'm not going to go into that, there appear to be mutations in certain regions of the DMD gene that lead to there being an immune reaction. So what they've done is to exclude enrolling any more patients with mutations in certain regions of the dystrophin gene in the clinical trial, at least for the time being, until they and regulatory agencies better understand what's going on. Now, this doesn't actually involve the FDA. It turns out the FDA hasn't signed off on the clinical trial, so no trial sites are open in the US yet. But one thing, in mentioning Pfizer, that they're starting to do going forward is to start running a small clinical trial involving non-ambulatory DMD patients, which is very important. Traditionally, just about all of the clinical trials in muscular dystrophies have been restricted to people who are still able to walk. But that leaves a lot of people out of it. So this is great news that they're extending it to include a broader variety of patients. So as far as Sarepta's trial, there haven't been any major safety issues, but there, was some concern about it not showing as much efficacy as one would have hoped or expected. And I think this is actually, well, what the company explained it as being due to is that they put people of different age groups. They had, people who were 4 and 5 year olds, meaning that they were past their 4th birthday, but not to their 6th birthday. And then they had 6 and 7 year olds, so they were past their 6th birthday, but not yet to their 8th birthday. And then they randomly chose, the people, half the people received a placebo, half the people received the real drug, and they compared how the two did. So in the 4 and 5 year olds, the people who received the real drug did better, but in the 6 and 7 year olds, both groups essentially were flat over a year, according to the NSAA test. So, after 1 year, whether or not they'd received the drug, they essentially had no change. But it turns out that just randomly, in choosing the people randomly assigned to different groups, the people who were given the treatment had more severe disease, meaning they had a lower functional score than the people who were given the placebo. And so I took this paper that was published a couple of years ago, looking at the trajectories of people with age who have DMD on this NSAA test. And what the left half of the graph is just all of the individual lines, and they divided people into sort of 4 groups, each quarter of the patient cohort in terms of who was progressing most rapidly, less rapidly, even less rapidly, and slowest. So the people who were treated in 6 and 7 year olds, I circled in this red circle here. This is the average trend line for all of the people in the most severely progressing quartile of the patient population from this natural history. And this purple line is the average age and average NSAA score of the treatment group in this trial for 6 and 7 year olds. And this shows where they ended up a year later. So they've essentially, no change over a year, but the natural history line is on a pretty steep downward slope. So the kind of the take-home message among all of the stock analysts was, yeah, they gave them this treatment and they didn't get any better. Okay, well, this is no good. However, taking someone who's on a severe downward trajectory and stabilizing them, I would consider great news. So to conclude this video, I think that the concerns with, the limitations as they currently exist of gene therapy and also some of the health risks are absolutely valid to look into. But what I would say is that overall, and there are exceptions such as X-linked myotubular myopathy, gene therapy, seems to be generally effective and generally safe in most diseases. So, I think it's important not to overreact. There are issues, there really is a need to make gene therapy safer, more economical, and better functioning. And there's a lot of people working on this, both within the neuromuscular disease research community and more generally. So anyway, that's the update for now, and I'm sure the new year will, bring a lot of new interesting things to discuss. So thank you for watching and wishing you well.